The monocyte macrophage system is also known as the reticuloendothelial system. It is a generalized phagocytic system located in all tissues, especially where large quantities of particles, toxin, and other unwanted substances are present. It is an innate defense mechanism which provides phagocytosis of the invading agents or either formation of antibodies and cytokines. So, the monocyte macrophage system is the combination of monocytes, macrophages, which may be mobile or fixed, and a few specialized endothelial cells in the bone marrow. The macrophages are found in the lining of the blood vessels in the bone marrow, liver, spleen, lungs, and lymph nodes. The monocytes constitute about 5.3% of the blood. The cells of the monocyte macrophage system originate from the monocyte stem cells. They enter the blood from the bone marrow and circulate for about 10 to 20 hours, after which they enter the tissues and become tissue macrophages upon stimulation. Their count is increased in infection, malignancies, and autoimmune diseases. The macrophages may be fixed in tissues or they may be mobile, where they are grouped together to form one big phagocyte to ingest the larger foreign particle, which may be the whole blood or the invading bacteria. The lifespan of monocytes in the blood is about 10 to 20 hours, whereas the tissue macrophages can survive months or even years within the tissues while performing their functions. The types of macrophages according to their locations may be the histiocytes, which are found in the skin and subcutaneous tissues, reticular macrophages, which are found in the lymph nodes, bone marrow, and spleen. Microglia are found in the brain, and Kupffer cells are found in the liver. Alveolar macrophages, which are found in the lungs. Beside these macrophages, others are the mesangial cells within the kidney. Only 15 to 20% of the mesangial cells perform the function of phagocytosis. Hoover cells in the placenta and osteoclasts in the bone. This is the origin of the monocyte from the hematopoietic stem cells. And these are the different macrophages within the bone, central nervous system, lungs, liver, connective tissues, and spleen. When the monocytes are stimulated due to any invading agent or pathogens, they are enlarged by different substances, protein molecules, which may be integrins, immunoglobulins, and endothelial growth factor. They cause the maturation of monocytes into the endothelial macrophages, which attach themselves with the blood vessels, and these integrins help them in growing and in cell-cell interaction, allowing them to pass through the endothelial cells by the mechanism of diapedesis. Once in the tissues, they may be resident macrophages and live in the tissues, which may be skin, liver, bone, and CNS, or they may be recruited macrophages, where they are activated by non-specific antigens, which are the generalized mechanisms of activation of macrophages, or they may be activated alternatively, means the macrophages are activated and help in the wound healing and tissue repair, or either the macrophages are activated classically, where they are activated by the certain microbial pathogens. Diaperesis is the passing of the macrophages while squeezing through the spaces between the endothelial cells. Then they move by the amoeboid movement. They are attracted by certain chemical substances which are released during infection or inflammation. And phagocytosis is the ingestion of the offending agent. The difference between phagocytosis by the neutrophil and macrophage is that the neutrophil can phagocytize only 3 to 20 bacteria, while macrophages can phagocytize 100 bacteria. These are the different steps of phagocytosis. While the invading agents are attracted by the phagocyte by chemotaxis, then it is ingested by the phagocyte. The phagosome is formed, which contain the phagocytic vesicles. Then it is fused with the lysosomes to form phagolysosomes, where digestion takes place. The indigestible material are vacuoled in the residual body, which are later on exocytosed. What kills bacteria by a phagosome? These are the lysosomal enzymes. For example, macrophages contain the lipases, which can digest the thick lipid membranes possessed by the tubercle bacilli, and other oxidizing agents of the peroxisomes such as superoxide, hydrogen peroxide, and hydroxylines. Why normal tissues are prevented from phagocytosis? They are prevented because they are self-antigens, they have smooth surfaces, they have protective protein coats that repel the phagocytosis, and another mechanism is opsonization. It is the process which makes the invading agent or the antigen more susceptible to phagocytosis. These are the diagram showing that the macrophage or the antigen presenting cells, they present the antigen to the cells, immune cells, which may be T cells. These are the different macrophages in the skin and subcutaneous tissue, which are known as the histiocytes. They form the first line of defense against infection, and you can see here these are the white blood cells within the skin fighting against any pathogens. The macrophages in the lymph nodes, if the invading agents enter within the tissues, they do not directly enter into the blood. There are the lymph nodes which entrap these invading agents before it enters into the blood. The sinusoids of the lymph nodes are lined by tissue macrophages. This is the structure of the lymph node. They have extensive macrophages and other immune cells such as B cells and T cells and plasma cells. These macrophages are lined by the capsule of the sinusoids of lymph nodes, where they are known as the subcapsular macrophages. The macrophages present within the alveoli, they are known as the alveolar macrophages. If the invading agent is digestible, the macrophages digest them and release the digestive products into the lymph. But if the invading agent is not digestible, the macrophages within the alveoli, they form a giant capsule around it for a time that it can be dissolved slowly. For example, it forms a giant capsule around the tubercle bacilli, the silica dust particles, or even the carbon particles. The macrophages within the liver sinusoids are known as the Kupffer cells. The invading organisms within the GIT, they pass through the portal circulation to the sinusoids of the liver, where the tissue macrophages are present in the form of the Kupffer cells. These Kupffer cells line the liver sinusoids and they entrap any invading agent enter within these sinusoids. You can see here these macrophages are lined here, and after which they leave the liver sinusoids and enter into the circulation. This is the portal blood vessel from where the blood comes, and if there is any pathogen, it is entrapped by the Kupffer cells and then it enters into the general circulation. This is another diagram. This is the portal vein. These are the liver sinusoids where abundant Kupffer cells are present, which entrap the pathogens before the blood enters into the central vein. The macrophages of spleen and bone marrow. If the invading organism succeeds in passing through the lymph nodes, it enters into the general circulation where it is entrapped by the macrophages of the spleen and bone marrow. In both these tissues, macrophages are entrapped by the reticular meshwork of the two organs, and when foreign particles come in contact with these, they are phagocytized. You can see here these are the marginal zone macrophages and periarteriolar lymphoid sheaths. These are the capillaries from the splenic artery. These minute capillaries, they directly fuse their blood within the red pulp, and from the red pulp, while passing through the trabecular meshwork, the invading organisms are entrapped within the macrophages within the red pulp, and then the blood enters into the venous sinuses. These are the venous sinuses. So the artery, from the artery the blood enters into the red pulp, and within the red pulp there are extensive network of macrophages which entrap the microorganisms, after which it enters into the venous sinuses. Osteal macrophages are another group of macrophages within the bone, and beside phagocytosis, these macrophages are helpful in bone modeling and remodeling. These macrophages are present near the osteoblasts and perform defensive functions. There are another mucosa associated lymphoid tissues which are populated by B and T lymphocytes, plasma cells and macrophages. They are present all over the body. For example, within the tonsil, nose, within the GIT and other lymphoid associated tissues related with the genitourinary system. The mucosa associated lymphoid tissues found within the small intestine are known as the Pierce patches. Here they contain the M cells, and these M cells are helpful in transporting the antigen from the lumen of the cells to the immune system, such as T cells. The macrophages within the tonsils, they form the Valdeur's ring and consist of the tubal macrophages, the pharyngeal tonsils, palatine tonsils and lingual tonsils. They all constitute the macrophages within the tonsils. The macrophages in the CNS are known as the microglia, and they serve as the defense mechanism as well as in developing central nervous system. They are important for the removal of unwanted synapses, and they define the neural network, helping in myelination and demyelination of the neurons during development. In this way, they help in improving the learning and memory processes. The mesangial cells of the kidney, these are the mesangial cells, and the only 15 to 20% of these cells are helpful to provide the macrophage system and phagocytosis, and whereas other cells they provide structural support and contraction. The hoof bar cells of the placenta, they also constitute the monocyte macrophage system in the fetus. They are present in the chorionic villi, where they not only help in phagocytosis but also they help in the vasoregulation by